All right, so I'm Ulysses, president of the MV Petri, and we are a 501c3 that I founded based in Virginia, but our activities are all throughout the DMV area. For ourselves, we offer science and tech meetups. And in fact, at this point now, we have about 10 different meetup groups that we've put into a large coalition down the DMV. So it's about 10 of us, and we span the gamut between traditional tech, biotech, entrepreneurship, startups, and I'm trying to think of the last one. Oh yeah, data and computers, all this sort of stuff. Okay, so other than that, we also have a variety of projects that we're doing down there. These are more of like community projects. Some of them span also tech, some of them span biotech as well. For example, we're trying to develop our own bio facility down in the DC area as well as a couple community projects that we ran last year. One of them notable for allowing us to publish an academic research paper based off of work that we did for a hackathon that was hosted by a nonprofit for a rare disease called neurofibromatosis. So in that sense, we worked on this project. We saw it through all the way to completion. It was about analyzing high throughput drug screening data, and then we actually managed to publish it. And it was published back in December of 2023. So we're very, very proud of that and showing the potential of what happens when you can bring over 100 different science and tech professionals from all throughout the DC area to work on things in their spare time. So the two slogans I use to describe the organization, the first one is we're helping people working on the next big thing in their basements, living rooms, and garages, just showcasing that my focus is on people way early in the development process for when they're coming up with new ideas or testing new things that they want to bring into the ether. And then the other one that I use is we do a mix of science education, scientific research, startup development, all in one nice science and tech community. So that's the slogan you're in there. Okay, so now back to the actual meet. So for this presentation, the title being Genetic Engineering and Neurological Diseases, I also want to acknowledge my co-writer for this presentation, who is Varun, and I have to remember to say it the right way, Srini Vasan. And he's a high school student who has been interning or volunteering, however you want to word it, and he's been a phenomenal resource for me and for the rest of the organization in helping us in all sorts of projects. So I'm very appreciative of the work he was able to do to get some of the research done while I was busy and doing all sorts of other random things in the area. So the general outline for the talk will follow three general points. We'll do some quick introduction to genetic engineering, just because of you never know who's in the audience, but for the people who probably frequent bugs, they're probably well familiar with it. If not, yeah, it's the general direction I'll take is more breadth than it is depth. So we'll cover these things in enough detail for you to follow along with the rest of the talk, but I'm not going to go down the rabbit hole of, that's deep into the subject. Then we'll go into looking in at some of the more popular neurological diseases that are out there that most people will hear about, and just giving the kind of brief descriptor for them. What is it? What are they? What's the issue? And then finally, we'll see the application of these things together, mostly in the context of clinical trials. So in terms of the introduction to the genetic engineering aspect of it, so the definition that you can grab from the National Human Genome Research Institute is that genetic engineering is the use of, or is the process of using laboratory techniques to alter the DNA makeup of an organism. Generally speaking, this is done in one of two general fashions. The first one of which we call in vivo, or doing it within the body. So in this case, for in vivo, what you want to do is, as you can see in this cartoon, right here in the bottom left side of the slide as I'm looking at it. And this cartoon is going to come up a lot in this presentation, just because I find it to be a really, really good cartoon, very, very good at just putting it all in one package. But if we look on the left side of the cartoon, we have in vivo genetic engineering or genetic modifications, which involves us taking a therapeutic gene, or in this case, in the form of a plasmid usually, or some other sequence, and then we're going to package it into usually what's a virus, but the general term for it is a vector. as we call it. So a transportation media. And then what we're going to do is we're going to insert it into the person directly from that point. And over at the injection part of this, you may see a lot of abbreviations in that corner. And all that is just medical abbreviation for the different ways that you can inject things into people. So you may see IV, intravenous, or inserting things directly into veins, IT, intrathecal, or the insertion of something into the spinal cord or brain, IM, intramuscular, so right here in the abdomen, and so on and so forth. The second general way that we do this is ex vivo. So in this case, instead of directly putting something into a person's body, we're going to take out some cells, we're going to put them into a Petri dish, and then we're going to follow that same initial process again of taking our therapy, packaging it into a transport media or a vector, usually viruses, but may be done in other ways, putting them into a Petri dish together with the stem, with the cells of interest, and then we'll put those back into a person. And so that finishes up that left-hand cartoon. Oh, there's something in the chat. Oh, what? No.
Oh, no, Libby says it's fine for her. Just for the one participant.
Oh, okay. All right. So then over on the right cartoon, we have a little bit more kind of the general fashions of how we kind of go about this. So you see that square there. So over on the bottom right corner, it's just another way of depicting the ex vivo gene therapy. But in terms of the other ways that we kind of will go and participate in genetic engineering, we have gene editing directly. So cut out, put in, gene knockout and silencing. So the insertion of something to bring down the expression, so prevent something from happening. Or if something is missing, like in the top left corner, then we want to put in something that's missing. So insertion, silencing, and then cut out or copy or cut and paste, for lack of a better word. So from there, so let's just say you're not, oh, okay, here we go. So in terms of those vehicles, those transport medias, those vectors, as we also call them, generally speaking, I mentioned they tend to be viruses. And this slide is just showcasing the various different viruses that people will use to get something into your body. The most familiar of these that you may see on the board here is adeno-associated viruses, or AAVs. They're very popular. Most people, if you have heard of them, they're in the context of some of the COVID vaccines that were released during the pandemic. So Johnson & Johnson most famously used adeno-associated viruses as their transport media to get the mRNAs into your body and then get that to produce spike protein, right? So the general mechanism there. Some other ones here, there's the kind of the big brother there of the adeno-associated viruses, just regular adenoviruses, simple retrovirus, not HIV, I promise you. And then lentiviruses, which I recall from my time in neurosciences, we use them there. And then herpes is new to me, using them as a transport media for genetic engineering. And then over on the right-hand side, what you'll see there is just a cartoon showing, a very, very crude cartoon showing how viruses can go and make their way into cells and then proceed to deliver your gene of interest or your modification of interest. So if you're not using viruses, though, that's okay. You can do it in other ways too. Well, like for example, once again, turning back the clock a few years ago, if you weren't using an AAV, you need some other vehicle to use it. In those cases, we were just directly injecting mRNA. But if you want to do some more complex genetic engineering, we have these mechanisms here. Most of us are familiar with CRISPR-Cas9, just because we use that a lot in the lab, but there were some other ones that were developed as well in the history of genetic engineering that will occasionally find use today. I won't go into depth much into the differences between those. Just know that they kind of work on the same general principles of breaking your DNA, and with the assistance of additional engineering for other things that you can package together with these mechanisms, that you can either get them to disrupt the gene by breaking it and then closing it in a way that doesn't completely close the loop, as you can see in the cartoon, or you can get them to correct the gene. So Once again, cut out, paste in the right one, or in this case, or in the last case over there, you'll see genetic insertion. So break and then put in something new. So that's kind of the overview, the quick and dirty on genetic engineering, at least from the way I tend to think about things. So remember, more breadth than depth in this case. So on part 2 of the talk, we're going to get into neurological disorders. So I guess for some background on myself, I spent 3 years doing research in neurological disorders from 2016 to 2019 over at NIH. And while I didn't study Alzheimer's, it's still an interesting disease anyways. But so Alzheimer's disease, just to give you the just because it's a little hard to see on the screen, but it can be described as the degradation of memory. So many people, unfortunately, will have some run-in with somebody who's had Alzheimer's. Most of us know them for loss of memory, or or in worst cases, loss of function because certain things can't be remembered, whether it's how to operate a piece of machinery, how to operate some sort of other thing required for daily living. As far as symptoms go, you can see there on the top right cartoon, you see things like confusion with time, difficulties with completing even tasks that you get familiar with over time, and other things such as judgment issues, emotional dysregulation, and so on and so forth. In terms of, big, large population statistics, at the moment, about 6.5 million people over the age of 65 are diagnosed with Alzheimer's. In terms of Alzheimer's sort of representation of general dementias, they do make up the majority of general dementia cases in the US, as far as I remember. In terms of what actually happens on a more molecular level, we have here in the middle cartoon this sort of, well, the top is a comparison with a normal brain, but in Alzheimer's, you'll notice as we move from left to right, you see just a general shrinkage of brain tissue, and this is because neurons or brain cells are dying, and they die from a combination of these two general accumulations of two proteins, one of which we call beta amyloid or amyloid beta, depending on how you were taught. And when these things accumulate in your brain, they can form plaques. So these hard collections that, if they get on the wrong parts of your brain, or the brain cells at the ends where the synapses are, or where the signaling is done between one brain cell to the next, if you get the plaques in there, it can mess up with signaling, or it can cause signaling issues. So messages don't get sent across your brain. And then in the cell body, so closer to the top of that cell there, you also see tau tangles, which can disrupt the sending of signals within a cell that tell the cell how to function. So moving on, the next kind of big one that many of us may or may not be too familiar with is Parkinson's disease. So Parkinson's disease is caused by the breakdown of nerves. The breakdown of nerves in many places, but primarily starts in a place in your brain called the substantia nigra. The substantia nigra is a very dark strip of brain cells in sort of the midbrain area. They call it the substantia nigra, but it's somewhere in the midbrain. You can see in the cartoon, if I zoom in a little bit here, it's very, very central to the brain. But that area has a lot of what they call dopaminergic neurons. So neurons that are very engaged in the production and sending out of dopamine to other places in your brain. And as you can see here, as the disease progresses, on the left-hand side, you have someone who has early Parkinson's, and you see that very, very dark strip, and that's like a color image. So that's just someone taking a photo of this. But as the disease progresses, you lose more and more neurons in that area. And what that means is signaling from that part of your brain to other places of your brain is completely disrupted.
What does the color mean?
No. I mean, they're dark in appearance. It's so it's in the name. So substantia nigra meaning dark substance. Okay. So that's kind of the They're neurons. So they're neurons that send out dopamine from their area to different parts of the brain. And dopamine, most of us know it as reward signaling, good feeling, but also has general function across the brain. So in terms of big numbers, about 90,000 people in the United States get diagnosed with Parkinson's every single year. Most of us know, or if you don't know, when it comes to symptoms, a lot of it has to do with motor control. So many people who have either dealt with Parkinson's or are familiar with it know it most from the motor symptoms. So things like tremors, things like hunching over, things like difficulty with walking, trouble swallowing. And then there are some other things that kind of precede those symptoms as well. So sense of smell, loss of a sense of smell is really common for a lot of the early stages for Parkinson's before you even see any of the motor symptoms, excessive drooling, and insomnia, actually. So people will, long before they get the motor symptoms, they'll actually have problems sleeping for a number of years before things get worse. So moving on, multiple sclerosis. So multiple sclerosis, a little bit, following saw a little bit of a differentiator here from some of the other ones. Multiple sclerosis is mostly a autoimmune disease. So in this case, it is the degradation of your neurons, not from a issue in the other senses where something is accumulating within the brain or something is being lost in the brain. In this case, another part of your body decides to attack your brain cells, and specifically a lot of your neurons that are not necessarily in the center of your like head. They have to do a lot more with some of your external neurons, peripheral neurons, as we call them, or across your body, arms and limbs. And in this case, oh, did pardon me. It looks like I forgot to put in the actual text there for multiple sclerosis. Yeah. Anyways, so I'm doing it all from the top of my head. So in this case, but the cartoons are accurate. So in this case, we have white blood cells that, for reasons not always really understood, your white blood cells will start to get a itching or a desire and think that your neurons that run through your arms and legs and outside of your head, generally speaking, are bad for you. And more importantly, or more specifically, they start to target what's called myelin sheaths. So they are these sort of fatty protective covering that goes over your neurons to help them improve with signaling. They act as insulation. So imagine, throughout your arms and legs, you have copper wires that help move electricity from one place to another. So signaling, and then to kind of keep it protected and make sure it works correctly. Like what we do is we cover our copper wires with a rubbery coating. Our neurons in our bodies will do the same thing. They get protected with a bit of a fatty coating, but in multiple sclerosis, that coating gets eroded because white blood cells start to think that it's bad for you. So you can see in the middle cartoon there, a kind of schematic of how this works. And just like many other, or more similar to Parkinson's, I guess in that sense, you see kind of motor loss, you see problems with like your arms and legs. So in this case, it manifests differently as a loss of balance, difficulty with cognitive function, muscle spasms, so on and so forth. So then next one being amyotrophic lateral sclerosis, otherwise known as Lou Gehrig's disease. The other name I'd come up with it over the time was Stephen Hawking syndrome as well, just because that's what he had. But this is actually the one that I spent a long time working on. So this is definitely one where I probably have the most knowledge of here. And but just to give some hard numbers, about 5,000 people will get diagnosed with ALS each year, and up to about 20,000 of them will have it at any given point in time here in the US. In this case, the cause of ALS is not really well known. all we have at this point is a description of what it is, and it's described as a very quick progressive loss of function in your arms and legs. So it starts with your what we call peripheral neurons, so arms and leg neurons, not head neurons. And as far as the molecular aspects of how this works, once again, we don't really know what causes it. We just kind of know what it looks like when it starts to appear. But by the time you get to that point, it's a little too late. So molecularly speaking, we see the return, or not the return of, but we start to see things kind of accumulate in these cells, in these neurons in your arms and legs. What accumulates in terms of things like proteins that start to build up in these cells can vary depending on what started it. So over time, there have been many different proteins that will build up. So SOD1 is one. It's a protein that helps with reducing reactive oxygen species. TDP-43 is one that normal function, we don't even know what it does normally. We just know that in this disease case, it accumulates in these cells. Let's see, C9 open reading frame 72. That's a protein that comes from, well, it's related to a part in your genome that's just like a really big extension on chromosome 9. Yeah, open reading frame 72. Anyways, and then FUS is another one that has to do with trying to reduce reactive oxygen species. So anyways, so you see a buildup of various proteins of an unknown or buildup of proteins due to an unknown reason, but the end result seems to be the same no matter what they are, is a really rapid degradation of your nerves that run through your arms and legs. And over here, all the way on the right-hand side, we see a cartoon just showing the kind of stages of ALS. And just to give you a time scale, you can go from early stage to late stage and end stage in from 90% of people, less than 5 years. So very, very quick. But yeah, lots of loss of motor function, walking, and then at the very end of it, breathing and swallowing tend to be affected. So anyways, so aside from that sort of grim segue, this is where the kind of merging of what we've been talking about so far come together in terms of like where they are in practical terms. So you have knowledge, and then eventually we have to do something with that knowledge, and we tend to express that in the form of clinical trials. So we come up with ideas, and then we see if they work by testing them in people eventually after testing them in other varieties. So, and this cartoon that you see here on the left, the bottom left-hand side of this screen or the slide, is one that's pretty much going to appear in almost every single slide going forward. And it's just a description of how we tend to type or describe clinical trials of different stages. So I guess just to go through that, before we start testing on people, we do a lot of laboratory studies, and the laboratory studies, or preclinical studies, as we also call them, can range everything from testing it in bacteria to yeast to mice to other animals such as other human primates, rabbits, whatever the case may be. Right? We have a lot of animals that we've kind of tested on in the past and will continue to test on, but most of the time the goal is to try and make sure that it works somewhere else before we try it on people. Then we have what's called phase 1 studies. So these are very small, usually in the number of tens. And the goal of testing our therapeutic after it's been tried in animals and cells is to see if it's safe on people. So in some cases, the people who are being tested in a phase 1 are actually healthy people. They don't have the disease of interest that you're willing to treat. They're just people who volunteer to say, hey, I also want to make sure that this drug is okay for people. But other times it can also be people who have the disease too. And this is just because in some diseases, they're either the disease is not one where, like in some case, severity, Like if the disease is really severe, it might just be better to try them out on people who are willing to volunteer for it. Or in some cases, like there are some therapeutics where if you know, not necessarily know ahead of time, but are suspect that it may be a little bit harsh around the edges, it might just be better to not use a healthy volunteer for it, but use somebody with the disease, if that makes sense. So there's a little bit of art to it, but generally speaking, we try to do it on healthy volunteers. But if we can't for any other reason, we will use them on people who have the disease of interest. It's just low numbers. Low numbers, make sure it works, make sure nobody suddenly collapses because they took this drug of interest. So then we have phase 2, and normally these are in the hundreds. So just doing more safety studies. And in many cases, these will be people who actually have the disease in a phase 2. And then phase 3 at this point is sealing the deal on safety, but also making sure that it actually works better than what's out there currently on the market. And the number for those is usually in the thousands. And then finally, we have phase 4, which tends to come down to long-term effectiveness, or sometimes they call them marketing studies as well, because sure, we did it on a couple thousand people in the phase 3, but if we expand it to another couple thousand, then we can really make sure or really say that it actually does work. And yeah, sometimes they're done for long term too, or follow up study as well. So let's just say, for example, we had a therapy of some sort, and phase 3 shows that it works really well, and then someone said, "Well, what does it still work 5 years out from when we did it?" And then you'll do that test, and then you'll say, "Well, it did, or maybe it didn't. But if it didn't, here's what we can do to try and extend the lifetime of it." So those are the various stages of clinical trials there. So anyways, so how did these end up get used, or how did we do these sort of genetic therapy trials in people who have Alzheimer's? So I'm not going to go through every single one. I thought even in my own kind of preparation for this presentation that would be a lot, but also B, there's no way I could hit a lot of things in a single presentation. So for all of these, I'm just going to go through 2. Usually one that was done pretty early on, as far as I could see, on clinicaltrials.gov, which is a great resource if you're interested in looking at clinical trials, whether you want to join one or whether you're like me and are just looking back at things that were done previously. So in this case, I have, in most of these slides going forward, you'll see kind of the title of the study and just some basic information about it. And then, yeah, just kind of going over it. So in this case, we have the study that was done in the mid 2000s, with the last update being posted in the late 2000s, just kind of them, documenting the last bits for their study. In this case, it was a phase 1, so 10 patients testing out the safety of it for early stage Alzheimer's. And in this case, they were given an in vivo gene transfer of neural growth factor. And over here on the right-hand side is just kind of because it was small enough, they just listed out each patient anonymously, who they were, how old they were, how many years have they had the disease, so on and so forth. So nerve growth factor is a protein that is made in the central nervous system, and its main point is to help grow new cells. In this case, because thinking back to what we discussed before, Alzheimer's will eventually lead to brain shrinkage due to a lot of cell death. The thinking here is that if we can find a way to maybe give people more nerve growth factor, maybe we can offset some of the potential cell loss that we're going to see later down the line as they progress. So we're going to give it to them in vivo directly into their bodies and see if that helps. So the results of that, at least in terms of the objectives of the study, safety being the number one concern, generally deemed safe. As far as I could see, nobody had any sort of serious effects. Nobody died after getting these injections. Nobody died within a couple years. And actually what you can see is for those that did seem to pass away early on after the start of the study. Uh, Normally, they'll provide detail as to what happened. In many cases, it's something else, or they had another complication, whatever the case may be. But most of the participants in the study did live for many, many years after these injections. What they also kind of showed, and there's only so much data you can get from these with such a low amount of people, but they did do some post-mortem analysis for those two that did pass a little bit earlier on the study. And what they found was there may have been some sort of recovery. And in the top right image there, all you see is where they were injecting the therapy. So I always forget I can do this. Yeah, there we go. So you can see that's kind of the red box there is where they were trying to place the original vectors that they were using to deliver the gene therapy. And this is just kind of them showing that you can kind of see it just because these markers here. So P75 neurotrophin receptor is just a marker that shows that there is something generally growing there. And then Nisi staining, I believe, was just a general cell marker, just showing cells in general. And then the other part that they did here was fluorescent labeling of cells. And the story that they're trying to tell here is that the red is tau. And thinking back to what I was showing earlier, tau is part of the disease process in Alzheimer's. So tau builds up in the body of the neurons and disrupts internal functions within the neuron. And NGF, the green marker, let me zoom back in, is showing that there should be, or more expression of NGF should indicate that these cells are also getting ready to kind of go growth again, some revival. That's kind of the thought there. And then finally on the overlay, two stories show up that out of three cells that they were able to identify in this particular photo, that two of them have both tau and NGF in the same cells, showing that even though there was tau in there, showing some disruption and some decay, that they're after injection, they're capable of expressing more NGF, meaning that in theory, they should be able to recover over time. Meanwhile, though, one did not. So that's kind of the story that they wanted to show there, showing that, shall they continue on with this study and try it in more people, maybe they might be able to see some more improvement over time. So that's kind of a more older study. I didn't see if they did any follow-up work on that one as far as I could tell. But looking at something more recent, so this one, as you can see, the most recent update was last year, November. And in this case, so this is a phase 1, phase 2. So in this case, they're combining two objectives. They want to make sure it's safe, but also trying to see if there is any sort of actual effect or as part of the main points of their study. In this case, they're looking at 15 patients with dementia or Alzheimer's. And once again, they're doing in vivo transfer of something called apolipoprotein E isoform 2. So in terms of why they switch, so in one case they're looking at something called nerve growth factor, and in this case we're looking at another called apolipoprotein. There were studies that were done later into, I want to say, I remember reading about apolipoprotein as early as like 2014, 2015, also in undergrad. And with apolipoprotein, it's a protein that helps move fat within cells. And in humans, we actually have 3 different isoforms that present in our bodies. Most people have apolipoprotein isoform 3, and that accounts for most of us here. Some of us have apolipoprotein isoform 2 being expressed, and some of us have apolipoprotein isoform 4 being expressed. If you have isoform 4 being expressed, you're much more likely to develop Alzheimer's in your lifetime. And like, depending on the estimates, you can see the max that I could find. One study said that you can be up to 20 times more likely to develop Alzheimer's if you have expression of this isoform. Now, because these are genes, you have expression and isoforms. These things actually, they come in pairs. So most people have 2 pairs of 3. That's the normal. Some people may have a pair of 3, a pair of 2, 2 pairs of 2, a pair of 3, pair of 4, a pair of 2, whatever, right? So you have combinations here. The worst of these is if you have both of your pairs of apolipoprotein 4. That's where you get the worst of it, that 20x likelihood of developing dementia. So on the other hand, though, if you have apolipoprotein isoform 2, you're actually less likely than the general population to have dementia. But you're more likely to have some other issues that I saw, like somehow you're able to be less likely to get dementia, but you're more likely to have cardiovascular disease if you have apolipoprotein isoform 2, right? Life is a game of trade-offs. So, so in this case, the thinking here is if we edit their genome to where they express apolipoprotein 2, perhaps we can recover them and send them to go the other way, because they're, at least in population studies, they're less likely to develop dementia if they have the second isoform, APOE2. And yeah, so that's the theory there. And then the right-hand cartoon is just showing the kind of the general mechanism of how they think apolipoprotein isoform 4 increases your likelihood of developing dementia. And all you really need to know is that the mechanism there being, they think it's because it leads to increased tau accumulation inside of your cell. So more of the red stuff that you see here in these pictures. So what did they find? So if they started this study, I believe they actually started it sometime in 2019. It's just the last time that they updated it was last November. Usually before you see actual results with pictures and investigate, like thorough investigation, like you saw with the earlier study, it'll usually be a couple years after they finish it. So they're expected to be done with this study in 2024, later in 2024. And so far, all they've shown through an abstract that they presented at a conference, as well as a press release from a company that a researcher was working on, or not the researchers, like the investigator for the clinical trial, that's their partner, is Lexio. They had a press release that they released almost 2 years ago now, showcasing that there was nothing that could be, no serious events that could be tied to the administration of the drug, meaning that generally deemed safe. And then as far as effectiveness goes, I mean, it's a press release, so they're not going to release anything bad. So at least for them, they're saying, well, most of the cohort is alive, and you'll see more from us in the second half of 2022. I did do some further investigation. They never had a press release later in 2022. So I'll let you make of that what you will. So then we'll move on into Parkinson's. So once again, that same cartoon comes up. I just like using it. And so once again, I'll be showing 2 studies, one earlier in the timeline, one later in the timeline. So in this case, this is a study that was done also in the early 2000s, the sponsor being a company called Neurologics. And the last update was posted in 2008. So the study's, been done for a little bit now. And in this case, they're doing a phase 1, so emphasis on safety, involving 12 patients with early stage Parkinson's disease. And in this case, they're doing, once again, so in vivo pops up again. So injection of a glutamic acid decarboxylase gene to improve the protein in that case. And just like in the other one, it's just showing the demographics of each of their patients, just general information. just to have it there. And as far as this sort of study goes, so the idea behind why they're trying to fix this particular gene that leads to this particular protein is you can see in the pathway here. One thing that I know I didn't cover is because I couldn't fit it in, but in this case, the reasoning here is that this particular protein leads to an increased production of GABA, and GABA is a neurotransmitter in your brain that primarily plays an inhibitory role. And one of the odder things about Parkinson's that, once again, I didn't cover, and that's my bad, but if we go back to the Parkinson slide for a sec, and we see the death of those neurons here, when I was doing like my grad school work, I found out actually that these cells don't die primarily because of decay. They actually die of what's called excitotoxicity. So they work too hard. They send too many signals, and then they just break. So just like flipping a light switch too many times before, the machinery breaks, or if you push any machine too hard, it's going to break faster. So that's what's happening to these cells. So the thinking here is if we improve the function of a protein that would lead to an increase in inhibitory signaling, maybe that could save some of these cells going forward. So that's kind of the logic there. And then once again, cartoon pumps up again showing in vivo. So in terms of how well did this work, and also remember, like, they're just primarily testing for safety. So once again, generally deemed safe. They did note that at least in some of the patients, and you'll see this here over on the right-hand side, that they did note some improvement compared to their baseline, or no, not compared to a baseline, compared to a untreated group. So they found another group of people who also had Parkinson's, more or less similar to their group, and then see how they tested clinically. So these are just scores of clinical testing. You don't need to know the details, just know that in this case, a higher score means more function instead of less function. But in this case, they're showing that their cohort that they treated with this gene therapy seems to have shown some improvement in being able to retain their function over time, over the course of a year. And then this middle image is just showing where they did some injecting of the gene therapy. So this is kind of implied, but I should probably say it out loud. Yes, they are doing these injections surgically. So I should just note that. Anyways. I don't know. I thought I should just say that explicitly. I don't know. Some people may have imaginations and say, well, maybe if I just inject into the arm, maybe it'll travel up to the brain. Sometimes they do. A lot of cases they don't. The brain is very privileged from many, many other things in the body. So one of the quick ways to actually get something into your brain is to open it up and put it in there. Yes, pretty much. Yeah. So the brain is pretty privileged from a lot of things. So it makes getting all sorts of therapeutics into the brain really, really hard. Not just gene therapy, but others as well. Drugs and antibodies. But that's a different discussion for a different day. Oh, oops. Dang it, hit the wrong button. Give me 1 sec. Will I get that back up if perfect time to use the bathroom, I guess. Give me 1 sec. Let me get this. Oh, share. And no, current slide. Okay, good. Is it back up? Excellent. Yeah. Okay, so let's see. All right, so the next one, more recent one, looking at Parkinson's. In this case, once again, so this is a phase 1, and I didn't write it on the text, but it is an in vivo study. And this is looking at 25 patients with late stage Parkinson's as opposed to early stage Parkinson's. So much more advanced progression there. And in this case, they're doing in vivo gene, so adding in a new gene for glial cell line derived neurotrophic factor. If you remember back to one of the other slides, we looked at another neurotrophic factor, and all that means is this is a protein that's meant to try and stimulate growth of neurons. So here, once again, because this is more recent, getting follow-up information is a little bit harder, but they did present some of their early results from the study at a conference in 2019 to, I believe it was like the European Congress for like gene therapy and therapeutics or something like that. Either way, showcasing that at least what they saw was once again generally deemed safe, and if anything, just a slight improvement in once that UPDRS, so clinical score, just estimating function, so your ability to be able to move and all these sort of things. So slight improvement, not so much, and we'll see where it leads later down the line. And the last update they actually posted was pretty recent, so that's the last time they at least reported back to their sponsors. What's up?
By bilateral seminal, are they referring to the area of
Yeah, where they put it. Yep. And yeah, okay. So then we're kind of moving here towards the end. So use in multiple sclerosis. So this is the first, at least in this showcase, that we're looking at phase 2 studies, or well, there was another one, but it was phase 1, phase 2 combined. This one's purely phase 2. So they're starting to take a look at effectiveness, at least in this case, for multiple sclerosis. So these patients were given a plasmid that codes for myelin basic protein. And you can see in the image here in the center that myelin basic protein is something that helps those myelin sheaths, so that insulative coating that goes over your neuronal axons, the part that's carrying signal away from the center of the cell to the next cell. It helps with kind of keeping the shape or keeping that kind of wound structure. So at least they're thinking, or there was evidence showing that like while they're being attacked by white blood cells, that they're unwinding. So maybe adding in more might help keep it more intact. That's the general thinking there. And then once again, baseline characteristics, that's just there for in case anybody really wants to look at that. But the end result of this was pretty mixed. So they reported that they saw differences in MRI imaging, but they didn't provide any MRI imaging. They just provided what they called volumetric data. So they take the MRIs, they put them into an analyzer. The analyzer has a formula for kind of estimating the size and three-dimensional shape of that particular brain structure. And at least on their reported data, they had said that people who received this therapy, at least in the sections that they were looking at, they seem to have been have more volume, meaning less decay. But once again, they didn't post the image, so make it that as you will. And then, so in terms of newer studies, when it came to anything after this one, I couldn't really find much that was actually being done in humans. And when I was kind of digging through some other papers trying to figure out why that tended to be the case, it looks like just as information kind of grows and develops, the thinking kind of shifted less from, well, what do we do to try and fix the decay in the neurons, to what can we do to get the white blood cells from attacking the neurons? So the big bulk in focus now is dealing with that. So there's less focus on genetic therapies for this disease and more about stem cells or modifying white blood cells, or the only other gene therapy thing that I could find, at least in my research, was this mouse study where they were doing genetic modification on white blood cells to try and prevent them from targeting these neurons. So And the reason, and this is just some immunology here, but they're modifying in particular not not the white blood cells that are directly attacking your neurons. Those would be called T cells, but they're looking at ones called Tregs, and Tregs are T regulatory, is what they stand for, and they tell other white blood cells like T cells to stop doing something. So they're an inhibitory. So in this case, the idea being, well, let's modify these Tregs. Maybe they're not regulating the actual T cells the way they're supposed to be to prevent them from attacking your neurons. And if we go back and we can find what's stopping them from telling their friends to stop attacking neurons, that could improve the outcomes. So just know that at this point, the field has shifted in multiple sclerosis away from neuronal decay and more towards dealing with the immune system. And then ALS, so my home disease. Very few, there were very few looking at anything genomics related. The ones that I did find, they weren't about modifying genetic code directly. They were more about kind of like what we did with the mRNA vaccines, is just giving people an external piece of mRNA or trying to silence something. And my interpretation from looking back at the field was it doesn't, I don't want to say it doesn't make much of a difference, but the figure on the right shows that only about 10% of all cases of ALS have some direct genetic familial component that you can easily find. The other 90% or 80% is of unknown cause. So we don't really know what's going on there. So you have to know more about the disease before you can try and target other therapies. And even amongst the minority of cases that are attributed to genetics, you can see that there's a huge amount of genetic variation there. So that's a lot of targets for a very small amount of that 5,000 per year. So my guess is without, if I just say it out loud, they're probably going to end up being orphanized. So many people will not look into it just because it's a small number. So that's kind of the end of that. That's all I got. Yeah, so there are not directly. So I didn't talk about this, but both ALS and multiple sclerosis use the same animal models in mice, and they're not directly the disease. They're called, the abbreviation is EAE, and it's like, I forget the actual like full name, but all it is a general mouse model of neurodegenerative disease. So it's not specific towards ALS or even MS. So in terms of even doing the preclinical studies in these two diseases, they don't exactly line up very well with the future human disease. So it makes doing research a challenge there, and the progress on those diseases will be pretty slow because of that. Any other questions? See? Yeah. So EAE models. So we used them in my old lab when we were studying ALS, and our friends were using the same sort of model. It's not a very good model, but no one's been able to figure out one that's closer in sort of the appearance of these two diseases. So until that happens, like I said, progress on those diseases will be pretty slow. And yeah. Any other questions about, anything, the diseases, the sort of kind of overview of how we do things, or clinical trials, or anything? No? Oh, okay, now I see some hands.
When you look at the studies, like when you're looking at how effective it was, did they ever look at astrocytes and whether they're sort of producing these growth factors, for example?
So for which diseases?
Or any of the ones that you just look at.
Yeah. So in my home disease, we did, and it was a more recent thing. So it wasn't until I think like 2017, I think, is when they started looking at astrocytes and other brain cells to see their contributions. So for those who may not be in the know, about 25% of your nervous system is neurons, which is the ones that send signaling. The other 75% roughly is supporting cells. And the way I kind of think about this is like telephone poles and wires. So your neurons are the telephone wires that send the signaling, but the rest of but the poles is everything else, and it's a support structure. So you're getting me into a tirade that I usually give when it comes to my own field is one of the weird things that we've neglected to do is imagine a scene where there's a bunch of telephone wires on the ground and your kind of technician comes up and he's looking at them and he's like, well, what's wrong with the wires? What's wrong with them? Why are they on the ground? We should fix these while they're still on the ground. And then your first thought is, did you not notice that they fell off the pole? Right? So this is the question of that. So yes, people are starting to look into these things more. There are two general like glial cells, as we call them, so supporting brain cells that are definitely getting more attention these days, and it's microglia, which is a type of glial cell that can be found in your brain that has a semi-immune function, so it can take sort of samples from things in your brain, head up towards the blood-brain barrier, and then kind of hand it off to the rest of your immune system to let it know if something is wrong. And then there's also astrocytes, which play a very interesting role in regulating signals between cells. So let's just say, for example, I have neuron 1 and neuron 2. And I want neuron 1 to send a signal to neuron 2 about 68% of the time or 65% of the time, not 100% of the time. So an astrocyte that's nearby, and I don't know how they do this, but they somehow know that in these scenarios, I'm supposed to send an inhibitory signal to neuron 1 and tell it not to fire at this point. Where in other times it'll back off and say, no, this is a good one. So astrocytes play a lot of this interesting role in like kind of high level or like trying to tease out those details about when we should be firing, when not, and if a signal is sent out from this one when it wasn't supposed to be received by the other one, I need to step in and make sure it doesn't. So how this is like well done in the brain, I don't, as far as I remember, it's not too well known. I studied these sort of cells more in the eye when I was just getting my feet wet at NIH. It's easier to access. So, but same thing. It's like, oh, well, how do they know? So lots of things to elucidate there.
But, sorry, my question is about like scaling up, especially if you're talking about, I'd like to think that humans are more complex than mice. But then also if we're using a very generalized, like induced neurological issue in mice, how do you actually scale it up to start like human clinical trials or?
As in, like, what work does it take to show that something in mice worked or is good enough to present to like a clinical trial? Easy. Well, not easy, but not easy. But when you see it, you're like, oh, that's easy. But no, you do a lot of testing in mice. So when let's just say, for example, we want to do a neurological test in mice, and aside from coming up with our model, which may or may not always be accurate to the disease, we do put them through a bunch of physical fitness tests, for lack of a better word, to try and estimate how well they can use their arms and legs in terms of estimating how well these neurons in our arms and legs will decay as a proxy. So like I remember some of these and you'll have to forgive me, some of these sound a little draconian, but like some of them resemble like putting them on a treadmill. Some of them have them like, oh, you put them on a wire gate, right? On like a wire grid, and then you let them hold on to it, and then you flip it over and then see how long they can hold themselves up before they fall. Or like some, yeah, like I said, some of these they're a little draconian, but it's us trying to figure out, can they still use their arms and legs in the case of like my research back in ALS, that's exactly what we were testing for, right? Like how well can they still use their arms and legs? So we need to physically fitness test them. And you'll do this in about anywhere between, yeah, for some cases where the person who wants to do a clinical trial or the approving agency, FDA or whatever, they may only request, okay, well, maybe you only need 35 mice to test it. Or in other cases, it'll be like, you need to test it on 250 to 1,000 mice before you, we think we're comfortable enough with allowing you to do it in humans. But yeah, so we have testing methods for mice that are both physical, but also because they're mice, we can also do other testing on them. So we can do things like cellular testing. So we can take a mouse, sacrifice it, get the pieces of brain that we need, and then see if there's decay after applying our therapeutic to them, and so on and so forth. So I hope that answers the question. Yes. Mm-hmm. Oh yeah, intranasal. Yeah, very you know, it's sort of, I don't know why it's experimental, but like it's not something that we think about a lot. Like for example, I don't have a good scientific answer for this. It's more of like a cultural one. It's if this makes sense. So for example, um when back in 2020, so on and so forth, I remember reading reports back then about people developing intranasal like vaccines for COVID, but they never got off the ground. But yet we proceeded with intramuscular injections, right? So the standard vaccines that we get, and even in my current field right now, so I kind of moved away from neuroscience and I work in HIV at the moment, and like, we do oral medications. We're experimenting with like 6 months injections of antiretrovirals or other there or monoclonal antibodies that you infuse into your body for like on a monthly basis. But there have been other people, and we have a researcher who's working on an intranasal like vaccine for HIV and some other stuff. And I don't know what why there's such a what an intranasal vaccine. Yeah. Hmm. So for some reason, it's just one of those things that it's either an afterthought or there might be some regulatory like burden that I'm not aware of, or just not enough people think of it at first. But I've definitely seen more than one case of somebody thought, yeah, let's do this intranasally. It might work better, and then it just doesn't go anywhere. I wish I had a better answer of like why you don't see more intranasal stuff. Is they'd rather get a shot than just like,
Like when you get like a COVID, like rapid one during drive-through, that's, I don't know.
I don't know, but did anybody here have allergies and like do like Flonase? Does anybody remember it being that bad? I don't remember it being that bad either. Yeah. I know the COVID tests were pretty bad, but that's because, they're like poking your brain practically, you know? That was my,
I need to make the argument that's why too, though, because the olfactory bulb like isn't as protected. And so if something went wrong during the injection, your lung would like have issues.
or in this case, it's most of the times they're nasal sprays, these intranasal like sort of trials. Well, it's more like a, right? Yours was a, yeah, yeah, with blood-brain barrier problems. That so much is true, but the weird thing is I just haven't really seen much of it. And I don't know if that's like an oversight or maybe sometimes what happens, this is just like digging into culture of research, right? But sometimes what happens is maybe somebody did try it like 20, 30 years ago, and the results are just that bad to where whenever someone looks into it, they're like, oh, they tried it, this group tried it 20 years ago, and it just really didn't work out well, so it's not worth pursuing. Like that happens. Um, I saw another. One of you two? I don't know. We can do both. It's fine.
Mine is a little bit more personal. It's So I have a condition where a portion of my brain wasn't formed at birth, right? And it people when they get in their 30s or their 40s usually develop cysts called have you heard of a rat cleft cyst?
No, I haven't.
Yeah, it's really, really rare, right? So when you talk about ALS and people now, it's very difficult for me to find information on it, but it really impacts like pituitary function and things of that nature. So I was just wondering if you had ever heard of it, if you know of any trials or studies around it.
I personally don't, but I'll like clinicaltrials.gov is definitely a really good resource for finding a clinical trial. Yeah, that's your best case of finding the clinical trial. What I can't guarantee is that you're going to find a clinical trial that's near you, because the website does take in clinical trials that are sponsored all over the world. So I remember just personal story on my end, I needed to get my wisdom teeth removed, and I didn't want to pay I didn't want to pay the first dentist that I went to his bill for it, and nor did I want to pay the next one after that. And anyways, so I kept shopping and like and I found like two clinical trials. One was in like Utah, right? And they were actually going to pay for it, like just pay for the travel and pay you to get them removed. It was all the way in Utah. I didn't meet the criteria, but and then eventually though I found after like a month or so, yeah, I waited a month. I found that actually NIH proper was offering wisdom teeth removal as part of a study. So I signed up for that. So, check the website. You'll never know what you come across. And yeah, if you have a condition now, or hopefully one doesn't pop up later, but if it does, check clinicaltrials.gov. There may be something that you can enroll in that would provide either treatment as it's done regularly today, or you get something in addition to it, but you have to know what you're getting into. So, and I did find out why the Utah study eventually would pay you for it, and it's because they were testing new analgesics, right? So anti-pain medication. So in this case, you were either going to get a placebo. So after your wisdom teeth, right? After your wisdom tooth removal, you're going to get a placebo, so nothing, or you would get an experimental anti-pain medication that you had no idea was going to work or not. So
But typically before it gets to like the phase 1 or phase 2, if they don't even have enough people to participate in like a lab study,
So with the phase 1 and phase 2, there is something that goes even before that, and this only works in the case of super rare diseases where the number is so low you can't do a phase 1, or if it's something brand new that hasn't been tried yet. And we had legislation that was passed for this. Actually, I should probably keep this because the microphone. But so we had legislation that was passed back in 2018 or 2019, I believe. It was called Right to Try. And essentially what it gave people with few options the ability to do was to sign up for like first in human and like pre-phase 1 type stuff, totally experimental, that they didn't clear the bar yet for phase 1. So yeah, so it's called Right to Try. And all that really means is if you find somebody, this would be like personal advocacy on your end, is you find a researcher and they're like, I have this medication, it did make it past mice, and the FDA is kind of wishy-washy about whether or not they're going to actually let me do this in a person. We can work together and do it under Right to Try. Yeah. Mm-hmm. Yes.
Yeah, did you see the one in the chat?
No.
There's a question about how are computational methods and machine intelligence being used in researching neurological diseases today?
Easy, barely. No, seriously, barely. In my I guess personal view as a scientist and as someone who's been working alongside clinical researchers for the better part of a decade, it is a relatively technologically conservative field where the scenario will happen of a data scientist, machine learning scientist, or someone presenting a really streamlined, high-tech, algorithmic way using computers to try and either diagnose something or to better identify something or to improve a methodology a little bit better. And it happens, they do their presentation, the results can look amazing. You know, our algorithm is 95% accurate, or it's, you know, has the same accuracy as a regular doctor, but it can do it in half the time. And then usually what will happen is a doctor or a scientist will come up and they'll go up to the microphone and they kind of cock themselves back a little bit and they go, but how does it work? And in machine learning and AI, there is a bit of a culture in the ethics of how they do these things, that they don't always look under the hood, or depending on the method that they use, unsupervised or supervised learning for these algorithms. If they're like unsupervised, they actually don't know, and the algorithm kind of trains itself or optimizes something based off of what it learns, and then it's up to the researcher later down the line to kind of validate, you know, what it's looking at. But like in terms of that, how the algorithm found its solution is not always known. So when the AI or machine learning researcher gets to that point, they're like, technically speaking, I don't really know how it's supposed to know. I just know the result. Then the doctor goes, oh, then I can't use it.
I actually work in data science and AI, if I speak a little bit of that.
Absolutely.
Like you've got these, you essentially have these black box discriminators. So you give it input and it comes up with an output classification of yes, this is that, or and yeah, I was saying like the person was asked was like, all right, what is it actually doing? There's no way to interpret a very complex machine learning model. No way. So there's nothing to learn from. There's no way for us to understand what the machine algorithm.
Mm-hmm. And because of this, the scientist or the doctor will say, well, I can't use it. And my fear is not because they don't understand it. As someone else who was a doctor tried to tell me, it's not because they don't understand it per se. It's because if they tell that to a patient, then, you know, what do you make of it? If I tell my patient, well, I don't know how it works, but, you know, I can tell you 95% of the time it's right when it says you have this disease, then the patient goes, well, I don't know how I came up with it, but I guess I have it, and I just have to go with it. At least with the way things are done now, even if it's slow, and the argument being, well, I can give you the steps that I took to get to this diagnosis, right? So either we can go back and double check the steps, or it is what it is.
A friend of mine is a radiologist in Pittsburgh. I talked to him about some AI tools for diagnosing scans. Works in trauma work there, and he says he's open to the idea of using it for like very obvious cases where he would look at a scan and just immediately say, oh yes, I've seen this 1,000 times, 10,000 times. It's
Mhm.
It's the cases where it's not clear if there's anything going on at all, and if there's something, and if there is something, it's something new or classifiable, which he does see on a regular basis. And that's when an expert opinion is essential.
Yeah.
But automating those very typical of your bone is broken here, here, and here, that sort of thing. That's something that I feel that understood, that it could be used, but
Mhm.
getting that accepted.
Yeah.
And practically used, it's got to take some time, right, for itself.
Yeah.
It's like also, at least I think there's like a joke in the medical field. It's like if you do a full body MRI scan, you're gonna find something wrong because they always abstain from it. It's like even the same thing, like pretty much all of us would have an MC4 virus, but it doesn't necessarily affect all of us in a negative way. And so if you look at a test and this is positive, it doesn't always mean that, you know, it has to, it's treated.
Yeah. So yeah, it's a really good question, and I think about it a lot now, because at least when I work with the MV Petri dish puts me at that nexus between technology and biology a lot. And the general relation, and I know it's recorded, but I don't care. I'm saying it anyways. The general relation is like people in tech are much more willing to go into biology and the biosciences than people in the biosciences are willing to go into tech. And this is something I see now over and over and over again. So,
Tech is all about automation. And if you've already mastered the things that technologists want to automate, why?
I see it as for speed, but
But yeah, that's the idea. Getting that trust.
Mhm. I agree. Huh?
It's also because the volume is lower, right? I mean, if you're getting a brain scan, it's something's wrong. It's not, like for mammograms, they do AI screening now, and that's been accepted because there's just thousands and thousands and thousands of them. But generally speaking, if you're going for a brain scan, it's because they're thinking something's wrong. It's not a routine thing.
That's true. All right, cool. Any other questions from the chat at least, or any last thoughts? Oh.
NGF could just be produced. Are there other delivery vehicles like the COVID vaccine? Are they still bothering to use viral vectors?
Viral vectors are used popularly, but there are other things that people are using. I mean, like we saw with the vaccines, right? Lipid nanoparticles, right? That's these ones didn't use it, but other fields, if you really want to see where all like the super high tech advancements goes, I kind of learned this over time. You jump over to cancer. They usually tend to try out things a lot sooner before other disciplines do. So, like for example, just one last example. I remember when I was it was 2020 and I was looking at, or 2020, 2021, and the new vaccines were going to be released for COVID. And it turns out, I remember coming across a paper that was written in 2018, and it was a review paper about mRNA vaccines and the pros and cons with like protein-based vaccines and all these sort of things, written in 2018 as a review in the field of, I forget which sort of cancer it was. It may have been breast cancer, it may have been a different type of cancer. But the cancer scientists were already like well ahead on trying mRNA vaccines for cancer-related issues years before we tried it out in infectious disease for COVID. So yeah, if you want to see the newest tech, jump over the cancer. You'll find it.
All right, terrific. Thank you, everyone. Thank you.
Wow.
It's been great.
Oh, thank you all. Thank you all for coming.
See you soon. Our next seminar is Tuesday, also hybrid. So we hope to see you soon.
Actually, I'll have one last slide up just as we kind of go. So for us, if you want to see more of what we're doing, if you're from the DMV area, if you're closer to DC, we are doing a series focused on bio for this year. By last year, I definitely spent it a lot more in tech, and this year is kind of a return to bio. So we'll have a series starting in March, and those are just the ones that I'll be presenting. I have 3 other kind of co-presenters who will be kind of rotating on the weeks. And then hopefully in May, we've been working on trying to develop our own biofacility closer to DC as well. So at least as a trial period, the community college down there has agreed sometime in April and May to let us do some hands-on workshops in their educational spaces. So yep, now I'm done. All right. Oh, Ulysses is fine. There we go.